It's a tremendous honor and pleasure to be here. Thank you guys so much for this opportunity. Thanks so much to the organizers for the invitation. Atul, I think, just did a really fascinating job of describing all the different types of things that can be done using the types of data that is already out there, old data put to new uses. With that said, new kinds of data are emerging every day, right? The human genome is a new way of thinking about how to map an organism. It's a kind of map, it's a kind of data, but new forms of maps are emerging all the time. And so what I'm going to try to do in this talk is just tell you about one emerging kind of data and the interesting things that it tells us about not the sequence of genomes, but how those genomes fold and why that's important. OK, so the human genome physically is just a long molecule. It's about 2 meters long. But it has to fold up to fit inside the nucleus of a cell, which is only a few microns wide. The question is, how does it manage to do that? How does it manage to fit inside the nucleus of a cell? This has been a very old open question. It's one that has particular urgency. The reason that it has particular urgency is that about 30 years ago, people realized that mammalian genomes like the human genome, they do this funny thing. The funny thing that they do is, if you look at, like a virus or something like that, you have genes, right? The instructions for a little protein machine, and then you have a promoter, which is right next to the gene and determines whether the gene is on or off. Right? So it's all very compact and well organized, right? It's sort of like, there's a button in front of your TV that turns the TV on and off. Funny thing about humans is we seem to have remote control elements, right? Called enhancer elements, right? They live very, very, very far from the gene in the 1D sequence, and yet somehow they seem to be able to turn the gene on and off. So initially people were like, that's impossible, that doesn't make any sense, and eventually people realized that was a real thing, and they named them enhancers, and there's this question of where the enhancers are. The enhancers make it really, really, really complicated to interpret genomes, because the regulatory elements are not where you would logically want to put them. How is this supposed to work? A basic idea that folks have had for quite some time about how this is supposed to work is that, well, the enhancer manages to turn on the gene by forming a physical loop that brings the enhancer into close proximity with the gene. So then the natural question becomes, can one figure out where the loops are? Can one reliably map the loops in the human genome, in other genomes that we care about? So that's the question. So I want to tell you a little bit about how to read genomes in 3D. This is really a field where there's been a tremendous amount of work over the decades, but things are just getting better and better and betterer. And so I want to tell you about some of the latest developments. So first, I want to kind of tell you about something completely different, which is the traditional way of looking at something, figuring out how it's folded, how it's geometrically arranged, is to, you look at it, or you look at it, say, under a microscope or through a telescope, et cetera, et cetera, et cetera. But this is not the only way to interrogate the structure of something. It's a very powerful way, and we heard some talks at this conference that have highlighted that. But it is not the only way. And a different way is, for instance, the way that Facebook does it, right, where they sort of map your contacts. Right? You can explore the structure of something, you can understand the structure of something by looking at proximity between its components. Let me give you a simple example. Most of you are probably familiar with this guy. Right? He lives in a fictional town of Springfield. That said, this guy, there's a whole show about him and the other Springfieldians. The trouble with the show, they've been doing it for so many seasons, it takes an incredibly long time to actually watch the whole show and kind of get what's going on. So, what do you do from a big data standpoint, right? It just takes a long time. Well, here's something you could do. You could start to surveil the Springfieldians. I'm surveilling the Springfieldians. Here I've caught him in extreme proximity with something he really likes, a donut. More events, right? Now I've caught this other individual that he seems to be interacting with. What's the deal with that? That seems suspicious. Here's more surveillance, more surveillance. Eventually I've filled a whole slide with all this surveillance, and I'm going to just take this raw data, the very data that you see on this slide, and I'm going to display it in a big data style. We'll make a matrix, right, which is just fancy for this, right? We're just going to look at how frequently the different individuals appear in events of extreme proximity with one another, right, in the same picture. And immediately, right, the big data reveals the structure of fictional Springfield. I know that this individual and that individual, they are up to something. And I don't know what they're up to, right? Because big data is not transparent that way, but they are up to something, right? I've discovered, a sort of a tool-style association, right? And whatever it is they're up to, incidentally, appears to have nucleated this whole family of events of extreme proximity. I don't understand exactly what that is, but I know that something is going on. Now, this is pretty much a total waste of your time, except for the fact that we can actually create exact maps that are exactly like this for the human genome using a method called Hi-C that I came up with in graduate school and developed together with collaborators at the Broad Institute and at UMass Medical School, and then together with my lab over the years. And the basic idea with Hi-C is we're trying to take pictures of little pieces of DNA of the nucleus, little photos. So like these two guys, they're cuddling. That's so cute. Let's take a picture of that. Let's take a picture every time that happens. And I don't want to get into the details of exactly how this works, but the long and the short is you can use, for instance, something like formaldehyde to glue the genome in place, cut it into little bits, mark the ends of the bits so that you can pull them down later, and then fuse the little bits together, right, in a proximity-dependent fashion using something called DNA ligase, right? DNA ligase, when it sees two pieces of DNA that are nearby, it'll fuse them together, right? If there's available ends, that makes this little circle. Now, the critical thing that I want to highlight, what has happened here? Here is an event of extreme proximity, but this is pretty hard to interrogate, right? Like Ed Boyden was talking earlier about how complicated it is to see what's going on in the nucleus. But now I've converted this event of extreme proximity into this funny chimerical sequence, right? A 1D DNA sequence that encodes the extreme proximity in a form that's really easily readable, right? Because we're very good as a species. We are very good right now. It's not news at the Future of Genomic Medicine conference. We are very good at reading DNA sequences, right? So this is something that I could read. So you cut this up into little bits. You pull out the interesting bits. You sequence the interesting bits. The first time that we made a map like this, we actually were able to read off 4 events of extreme proximity, and I was like over the moon about the fact that we could read 4 events of extreme proximity. These days in my lab, we produce maps like this where we map 10 billion events of extreme proximity. And at that point, I would say it's fair to call it, well, bigger than 4. OK, so I want to give you a tiny bit of sense of what this data looks like. Again, it's just the matrix, right? It's the same thing we were doing with the Simpsons, right? The human genome comprises 23 pairs of chromosomes. Any locus, any position in any of those pairs can, in principle, form contacts with any other locus in the human genome. And so here's the map. There we go. And immediately, some structure actually jumps out at you, right? You can see these squares, right? How many squares are there? There are 23 squares, right? Why are there 23 squares? It's because we have 23 pairs of chromosomes, right? Each chromosome lives in its own little territory, its own subvolume of the nucleus. It doesn't really interact with all the other chromosomes quite as much. And so that's why you see these bright squares. It's not, if I'm on chromosome, I don't know, 19 or something like that, I spend most of my time hanging out with other bits of chromosome 19, not, chromosome 1, right? It's kind of a different part of town. OK, that's not news. That was sort of hypothesized in the 19th century, demonstrated through beautiful work by the Kremer brothers, the 60s and 70s. Right? That's not news, but it is a nice sort of positive control that these things are meaningful. Let's zoom in, right? This talk is really a story about zooming in. Here is the map of chromosome 8, right? And you can see, right, the more contacts you have between a pair of loci, the more often they're physically proximate, the brighter it is, right? Along the diagonal, right, you're seeing bits of the genome that are adjacent in 1D. They're nearby in the chromosome, right? And by virtue of being nearby in the chromosome, they'll never get very far from one another in 3D. So they're forming lots and lots of contacts. Whoops. They're forming lots and lots of contacts. Right, so this is good, right? And there's some complicated structure there. And I'm not going to get into the full details of the complicated structure that I'm showing because I don't really have the time to do that. But the point is that we can create these 3D maps, and they tell us something about which bits of the genome are close to which other bits of the genome. So now let's go back to this problem of mapping the loops, right? We're trying to figure out where the loops are, and the loops should look a very specific way in a Hi-C map, right? So let's suppose these 2 bits of the genome are in close physical proximity. They tend to form a loop, right? What should that look like in one of these maps? It should look roughly like this, right? These 2 bits have enhanced physical proximity. As I move away from either of them in 1D, the physical proximity should diminish. So I should see a bright local peak in the matrix. Good. So that's it. And in fact, it takes a whole bunch of effort to see those peaks. And I really, the stuff that I'm talking about now is work that was done in my lab, led by Suhas Rao and Miriam Huntley, who did the amazing job of actually trying to kind of get this to really work in practice. And the problem that they ran into is, so here's the very first Hi-C map that we published. And you can see the chromosomes look great. But when you sort of zoom into the scale of individual genes, so a gene is roughly the size of this box, right? Each time I'm zooming in by a large factor, right? So each time I'm zooming in, here's a whole chromosome. This is roughly the size of a gene. Now you can see the data looks beautiful, right? It makes a big data person very, very happy when you look at this scale. But at this scale, it's just sort of noise. It's just this, bunch of points, and they don't seem to have much structure. It's pretty crummy. And there was a whole bunch of effort to improve the quality of these maps, but the general paradigm was still the same. At the chromosome scale, it looked beautiful, but at the scale of individual genes, right, at this little box below, right, it looks pretty crummy. And then finally, actually, by changing a whole bunch of things about how we were, the quality of the data, how we were generating, how we were doing these experiments, we started to do these experiments instead of blowing the cells apart. We started to do them in intact nuclei. All of a sudden, folks in my lab figured out that they could make maps that were of much, much greater quality. So here's an example, and there's about a couple of dozen of these maps that are now publicly available, right? Start at the scale of a whole chromosome, right? Chromosome 7 looks good. You zoom in, zoom in, zoom in, right? Here, every pixel represents all contacts between 500,000 letters of the human genome and another 500,000 letters. Down here, right, each pixel represents all contacts between 1,000 letters of the human genome and another 1,000 letters of the human genome. So there's 250,000 little tiles like this and each tile like that, right? And there are 23 squared tiles like that, right? Because this is just a single chromosome. Well, the nice thing is, of course, that once we finally reach the land and the scale of individual genes, the loops become very, very, very obvious, right? Anyone can see them. And this is good because for years after the initial development of Hi-C, despite the fact that it was designed in order to map the loops, no one could see peaks that looked like this. And now all of a sudden we could see them everywhere we looked. So I just want to give you a bit of a sense of what it is we're looking at. I'm going to show you a lot of these maps, so I'm going to take the time to just explain exactly what we're looking at here. So this is a little portrait of 400,000 letters of the human genome. Each pixel represents the frequency of contact between 5,000 letters of the human genome and another 5,000 letters of the human genome. And you can see that it's elevated in some places and lower in other places. The number of contacts at which we sort of assign maximum redness is 193. Right, so the point is we have lots and lots and lots of contacts in this individual bit of the human genome. And lo and behold, there's a loop there. And that's very easy to see. And in fact, everyone in my lab knows, and I actually strongly sort of believe this principle of big data analysis, which is that you should just be able to look at the data and just see what's going on. So everyone in my lab knows that the gold standard for whether a loop is there or not is I print out the contact map, I bring it to my son, at home, and I say, and we started doing this when he was 3 years old, and I say, Gabriel, can you find the loops in this map? And he just points them out, right? And if he points to it, then you know it's there. And if he doesn't point to it, your algorithms, your life, your theories may have a lot of beliefs, but it's not there unless a 3-year-old can see it. And that's really the standard. So, of course, how do you map loops across the human genome? It's a big problem because the number of pixels that we have in these maps at highest resolution is larger than the number of pixels that Google Earth has in an image of the surface of the Earth. Right? So it's a lot of pixels to sort through. So clearly the right thing to do is you print out all these maps, you bring them home to your kid, and you say, look, you better circle all the loops or you're not going to get dinner. And I spoke about this with my wife, who didn't feel the same way about this issue. And we hit on, I think, an effective compromise. On the one hand, my lab developed a machine learning algorithm to just try to map the loops, kind of an automated fashion. On the other hand, we now have 4 kids. So, a little faster. Anyway, the point is, in the lower left, I'll give our little algorithm its opinion, right? Here it states the opinion that it thinks that there's a loop there. If you disagree with our algorithm, though, you're right, and the algorithm is wrong. And that's kind of my fundamental philosophical view on the matter. Whenever it states that there's a loop there, it's going to tell you how many contacts there are under the peak. So here there's 281 contacts under the peak. 281 photographs of those 2 bits of the genome close to one another. 281, the point is that's not 2. It's not 3. It's not 1. It's a big number. It's kind of the number you can do statistics with. So not only can your kid figure out that this thing is there, but your computer can usually do it too. OK, so there's a loop. Here's another loop. These things reproduce all day long, right? So here's a primary map, the replicate map. You can see they look pretty much the same. The big difference here is these numbers are a little bit lower. That's because we did 3 billion contacts here and 2 billion contacts there. Things that are supposed to loop should overlap one another under a microscope. And lo and behold, they do overlap one another under a microscope. So that was good news. In general, here's a striking thing. So whenever you see a loop, you end up seeing what we call a contact domain, this bright square. You've seen this before, right? This individual and that individual are up to something, and whatever it is they're doing is causing enhanced contact frequency across a whole interval of the human genome. Now that's striking, because you have a point-to-point interaction between 2 bits of the human genome, but that point-to-point interaction can modulate the frequency of contact across long stretches of the human genome, millions of letters of the human genome. Now these loops, are they interesting? Well, they are, I don't know on some level, but I can tell you that they're interesting enough that evolution has been kind of keeping them around. Let me give you some examples. So first, if you look at the loops across many human cell types, they're frequently in the same position, right? There's one example. Here's a fancier looking example. And actually, half the loops that we identify in mouse are present at the corresponding position in human. So this fold, right, is at least 60 million years old. That's an interesting question. So that suggests that they're doing something. And let me give you a sense of what we think they're doing. So here's a boring 3D structure. Let's look at the gene ADAMTS1 and GM12878. It's a lymphoblastoid. It's an immune cell type. And the gene ADAMTS1 is off, right? No RNA-seq signal. Very boring 3D structure. Now, let's look at IMR90 lung fibroblasts, right? Now ADAMTS1 is on, and there's this much jazzier 3D structure around it, where actually the promoter region of ADAMTS1 is engaging in 6 different loops with different positions in the genome spanning over 2 million letters of the genome. So you can see loop after loop after loop after loop, and now it's on, right? So somehow these loops, and we see this time and time again, there's an incredibly strong association between the presence of loops and dramatic activation. We're talking about 20-fold, 30-fold, 50-fold activation of a gene in the presence of a loop, where it's off, in the absence of a loop. And so what that's telling us is that as genes are turning on and off, the genome is folding in different ways, right? So why is a heart cell that beats doing something different from a brain cell that thinks they have the same genome? Well, it's because different genes are on. When different genes are on, it's because they're folded in different ways. So in some sense, the genome has this form of origami, right? You take your paper, you fold it one way, it's a paper hat, right? Another way, it's a crane, right? Same thing with the genome. You fold it one way, it's a heart cell. Another way, it's a brain cell. OK, so I promised the code. So here's a code. Here's a funny thing, right? We'll start with the funny thing before the code, right? The funny thing is, when you look at the anchors of these loops, the positions that are actually looping, they're pretty much always bound by the same set of proteins, right? One of these proteins is called CTCF, right? And people had thought that maybe CTCF was involved in at least some loops. And the other one is these, you see subunits of called RAD21 and SMC3. They're subunits of a ring-shaped cohesin complex, which is known to be able to associate with DNA and kind of, and be able to envelop DNA, but it was thought to do this just during metaphase. Anyway, you see these things time and time and time again. OK, that's interesting. It's not totally inconsistent with what people thought. But here is something which completely convinced me that someone in my lab they didn't really know how to code. I think that's the problem. That was my interpretation. So here's a funny thing, right? So CTCF, cohesin doesn't really bind directly to DNA. CTCF does bind directly to DNA, so it's kind of guiding the cohesin to its spot. And we'll talk more about that in a moment. Funny thing is, CTCF does not bind to DNA willy-nilly. It only binds to a particular DNA word that's about 10 letters long. Now, DNA has 2 strands. You think of the Watson strand, Crick strand. That means any motif, any DNA word can point either this way or that way. Now, that means if I've got 2 DNA words, right, like my 2 CTCF sites, they can either be pointing, both this way, both that way, away from one another. We call that the divergent configuration, or towards one another. What we call that is the convergent configuration. So he started to look at whether the words, had some sort of orientation bias. And what was really striking is, so he looked at these words and said, well, you know what? Every single time the word that's upstream is always pointing forward, and the word that's downstream is pointing backwards. They seem to point at one another. And I said, that's just silly, right? They can't possibly be doing that. because they're millions of letters apart. It's 10 letters long. Why should they care? It doesn't matter. It's completely inconsistent with, philosophically, everything that we think about how these loops form. And there's just a bug in your code, and you really shouldn't bother me until you debug your code. Anyway, in hindsight, that was a very embarrassing little, at the time it sounded great, but in hindsight, that was kind of an embarrassing little speech to have given. Because this kind of really works, actually. Pretty much like over 90% of the loops, they obey this convergent rule. And we actually now think that these other ones, they're just places where computational algorithm just made a mistake. So we think that actually the loops, and this is a real bit, 2.1 MB of the human genome, is a real interval of the human genome. And you can see, right, each of the loops is anchored at these convergent CTCF motifs that are pointing along the barrel of the motif. And now we actually like this so much that for my birthday, my lab actually rendered this bit of the human genome using cupcakes. Each cupcake represents 10,000 letters of the human genome and about 400 calories. This is almost certainly the unhealthiest genome browser in existence, but it has other merits. Anyway, so the last thing I want to do, just as I'm wrapping up here, is I want to tell you how is it that this actually works, right? This convergent rule is bizarre, and this is actually something that was sorted out by Adrian Sanborn and Suhas Rao, two incredibly talented students in my lab. And here's the philosophical problem, right? Everyone knows how these loops form, right? It's supposed to be, you have one bit, one looping element over here and one looping element over there, and they find their binding site, and then the genome wiggles and wiggles and wiggles, and eventually they're in close proximity and they bind. And this is this sort of model that loops form by diffusion, and this was kind of what everybody thought. But the problem is this, which is how in the world do you square that with the convergent rule? Because that implies that somehow these two little elements somehow know whether they're pointing at one another or pointing away from one another along the code of the genome. Now, if you blow up if you blow up a protein by a factor of 1 million, you blow up the involved proteins, right? You blow them up by a factor of 1 million, right? With super duper expansion, methods from Ed, right? Then they'd be each protein is about the size of a pea. The contour length of the loop is a kilometer, right? So somehow these two peas are supposed to know which way they're pointing, despite the fact that they're separated by like a kilometer genome. That doesn't make a lot of sense. And yet we always see them in this configuration rather than that configuration. So we thought we need a theory. So here's the theory we came up with. We had no data whatsoever. This is not big data. This is a no data theory. Our idea is that there's two donuts, right? Two of Homer's donuts. They tether to one another and bind to the genome at some random position. And then they start sliding in opposite directions. Again, there's no data whatsoever for this. Now, furthermore, these donuts, as they slide, they're constantly looking, because eventually it's sort of boring to drive, forever in one direction. Eventually you stop, and I like to stop at stop signs. These guys are constantly looking for a stop sign. And what's the name of the stop sign they look for? It's the CTCF motif. But of course, I don't stop for any stop sign. I only stop for stop signs that are pointed towards me. Otherwise, they don't affect me, right? So this guy, he hits it, he stops. Oh, there's a stop sign, but it's pointing the wrong way. I'm not going to stop. Oh, here's my stop sign. I'm going to stop for that. Now, there is no data, right? This is the opposite of big data. There's absolutely no data whatsoever to support this. However, right, if such a thing existed, then it would explain the convergent rule. So there's a tiny, 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 tiny bit of data to support this. OK, last thing I'll say and then just wrap up is if any of this stuff, well, either all this stuff is nonsense or the following experiments should work, right? If we understand the code of the human genome, we understand which way the CTCF motifs have to point in order to establish the loops, then we should be able to manipulate them and kind of control what's going on. I just want to show that that is in fact the case. So here's a prediction, right? We do this using simulations before ever doing an experiment, and then we do experiments to map things. This is a particular kind of region of the genome we call a transitive triple. It looks like this. A loops to B, A loops to C, and B also loops to C. You see tons of these in the genome. And of course, according to the convergent rule, A needs to have a forward CTCF because it's looping to two downstream loci. C needs to have a reverse CTCF because it's looping to two upstream loci. And B has got to have CTCFs in both directions because it both loops in both directions. And so that's exactly what you observe. So now you can just play, right? So let's get rid of the CTCF motif at A. If I get rid of the CTCF motif at A, I should kill the two loops that are anchored at A and nothing else. And we're going to do this using ultra-targeted mutations. These are like 20 base pairs long. We're hitting that CTCF motif and nothing else. Great. Great. Well, we do that, and lo and behold, that's exactly what happens, right? The loops go away. It's exactly as predicted. OK, but this didn't test the convergent rule because there was only one CTCF there. Now let's go to B. Let's test the convergent rule. We're going to get rid of the CTCF motif at B that points backwards. That should kill the loop from A to B, but it shouldn't affect the loop from B to C. And lo and behold, you do that, and that's exactly what you observe. OK, now let's have more fun with the convergent rule. Let's get rid of B forward. Get rid of B forward, you should lose the loop from B to C, and in fact, that's exactly what you do. You lose the loop from B to C. You don't lose the loop from A to B. So here we said, let's not harm any base pairs in the human genome. It's not ethical to harm base pairs. Let's not harm them. We're just going to flip them around. So we're going to take a stop sign. We're not going to chop it down like we did before. We're just going to turn around 20 base pairs in the human genome, make them point the other way. Now, if orientation doesn't matter, this should do nothing. But if orientation does matter, then all of a sudden that B to C loop should go away. And in fact, it does. The B to C loop is now gone. OK, well, for the maximum possible amount of fun, you can get rid of this stop sign, but then flip that stop sign around. And as a result, you should save the A to B loop while simultaneously getting rid of the B to C loop. So we're going to combine both of those. And lo and behold, that's exactly what happens, right? By just deleting this thing, but then I flip that guy, right? So if you compare these two, they have the same deletion at the B reverse motif, but this guy's got the flipped B forward. And in comparison to here, where you have the B to C loop, but no A to B loop, here you've got the A to B loop, but no B to C loop. And you're doing that just by flipping 20 base pairs, not even getting rid of anything. So we have kind of total control over this business now. If you are unhappy with how your genome folds, if you want to, for instance, write your name in your genome or other stuff like that, this is basically doable using a garage biotech. No fancy reagents here other than amazing CRISPR reagents that obviously make a lot of this stuff possible. So in conclusion, we can now map the loops genome-wide. They tend to form between convergent CTCF motifs. It's like a 3D code in the human genome. The data is consistent with loop formation by extrusion, although there's almost no data for that. The data for that is improving, though. And by modifying CTCF anchor motifs, we can actually add, move, and delete loops at will. Anyway, I'm funded by these folks, but I'm funded by fewer folks than a tool, so maybe I could read them with more hope. But I'm not going to try to do that. But I mostly want to tell you that I mostly want to tell you that Suhas Rao, Miriam Huntley, Adrian Sanborn are brilliant students who pretty much did all of this work. And I am honored to have the opportunity to present it to you guys. Thank you so much.